Okay, maybe now this works. So first of all, I would like to thank the organizer for the opportunity to give this presentation, in which I will talk about a very recent work just started a few months ago with Marina Bertocchi of the University of Bergamo and Ettore Mosca, Roland Reibol, and Eliana Zucchi of the Institute of Biological Technologies in Milan. So in this talk I will talk about the chromatin structure of pluripotent stem cell, and in particular I will propose two simple geometric models, like toy models, that we can use in order to evaluate the state of stem cell. In particular, why to study the chromatin structure? Because it's very important in order to understand the different function of a specific cell. In the human genome we have a very large quantity of genes, but the identity of a specific cell is identified by the gene that are active. And so in order to understand the function of this gene, it's important to understand the structure. On the other hand, also the state is important because it can be diversified in open or close. Open in the sense that is open to the transcription of some genes, and close that is transcription of the signs. So in this recent work we have first of all introduced two simple geometric models in order to describe the chromatin structure. And the assumption that we made is that we consider a solenoid model in which a single turn of a solenoid is composed by 6 nucleosomes. The radius of each nucleosome is 5.5 nanometer. The wrapping angle around a nucleosome is 3.5 pi. The peak of the helix is 2.5 nanometer, and the nucleosome wrapping length around each nucleosome is around 147 base pair. So in particular we have also fixed a constant length between each nucleosome, each pair of nucleosome, that is fixed to 50 base pair, such that the total, the sum of the wrapping length plus the linker DNA length is around 200 base pair. So the aim of this first part of the work was to propose a geometric model, a parametric equation that describes such a kind of structure. So first of all I will propose this simple model in which the 6 nucleosomes are represented by this parametric equation. So xi represents the I nucleosome from 0 to 6 to 5, which represents the 6 nucleosomes inside one single turn of the solenoid. And here the parameter xi that appear inside the parametric equation represent just like an arc length around the curve, and all the other parameters, so pi P is fixed from before, is the pitch of the helix length, theta is the turning angle, and so on. So all the other parameters are fixed from before. In particular, we have normalized every component by this quantity, so by the length function, such that every nucleosome have constant length equal to 147 base pairs. So this is a top view, and this is a side view of the same conformation. So the idea is that to connect by cubic Hermite spline the 6 nucleosomes according to this equation. So we consider this linear combination of Hermite basis function, which are given by this function, where A00 A11 represent respectively the initial and the final point between two successive nucleosomes. A01 and A10 represent respectively the derivative at the beginning and again at the end of the linker. So by this parametric equation we can also describe the connection between nucleosome X0 and the nucleosome XY. Again, xi represents the parameter along the curve, and so every connection is parameterized in the following way. in this way, we can obtain the final curve, which represents a single turn of the solenoid, and again we can see a 3D view and a top view. In particular, is here represented also the connection with the layer that is that is translated up in the z direction of 2 times the radius of the solenoid. So this was the first model that I call 6 helices. The second one is much more simplified. So we suppose to model one single turn of the solenoid by a torus unknot that is given by this parametric equation, where again psi is a parameter along the curve, and capital R and small r represent respectively the big and the small radius of the torus where the knot belong. Q and P are 2 parameters which tell us how many times the curve travel in the longitudinal direction with respect to the meridional direction. So in this simplified model, because we want that around each nucleosome the DNA turn twice, then we fix Q equal to 12 and P equal to 1. Again, the length of the Fourier torus unknot is normalized by this quantity such that the length of every turn of the solenoid has constant length equal to 1132 per pair. The final model of the solenoid is then obtained by connecting again by cubic spline function each single level of a solenoid with another one. So by connecting by cubic spline we have a final structure like this. Here is just a simple example of 10 levels where every color represents one different level, and again here we can see a 3D view and here a top view. So what we have done was we have first of all compare the 2 models in terms of geometric quantities, and then we have applied this model to some data, cheap data for pluripotent stem cell. So I briefly recall the quantities that we are going to use. We have considered the normalized total curvature, where it's given by the integral along all the curve of the curvature divided by the length, the normalized bending energy, the square of the curvature divided by the length, and the normalized total torsion, again the integral of the torsion divided by the length. So this is a very preliminary work, so I've just considered these quantities, but we would like also to include further measure of complexity like the Reiding number. And then let's see which the data that we have used for this comparison. We have considered cheap data, that is chromatin immunoprecipitation data, which consists in an experimental strategy that is adopted in order to find the open and close portion of DNA region. And so it allows to understand, to find which parts of the DNA are much more condensed versus the one that are less condensed. So to find the heterochromatin respect to the euchromatin. In particular, the data that we have considered are obtained are reported in this paper by Boyer et al. Published in 2005 in Cell, where they consider data obtained by specific antibody against this free transcription factor OCT4, SOC2 and NANOG that are very important for pluripotency. In particular, they have proven that they co-occupy the same portion of region that are related to the pluripotency. So in this Venn diagram we can see that the region, the area that are occupied by the free transcription factors SOC2, OCT4 and NANOG. So we have developed an algorithm which includes the free transcription factor region, bounding region, and the target genes that are affected by the free transcription factor. And then we have a compute for all the 23 chromosomes, the open and the close base pair. In particular, we can see in this second graphic the ratio between the base pair close with respect to the open, and these data refer to just the base pair and the genes that are affected by this free transcription factor. From this graphic and from the numerical result, we have found that the chromatin state is not uniform in the pluripotent stem cell, and the condensation of chromatin structure is obtained in chromatin 19, in chromosome 19, and in 22. In this figure, we can see the spectrum of open and close base pair by the free transcription factor activity, and so we have just included in this case around 3,000 genes. So we have then compute, according to the previous model, the total number of solenoid layers in a very simple way. So we have compute the total length where the chromatin is close, then taking into account the fact that every solenoid length is costal length, then we have obtained the total number of solenoid layers, that is of course proportional to the length of every chromosomes. And then we have also computed the three quantities, the normalized total curvature, torsion, and bending energy, and which again reflect the fact that chromosomes 19 and 22 are the more important for pluripotency. So in conclusion, we have proposed two simple geometric models for chromatin fiber conformation, and we have tested them on chip data generated for pluripotent embryonic stem cell, and so the model provided insights into the chromatin structure in terms of geometric information. In particular, the main result is that we found that the chromatin state is not uniform, and the more decondensated chromatin structure by means of a free transcription factor is obtained in chromosomes 19 and 22. So here there are some references related to the work that I just described. In particular, the data that I've considered for the computation are from this one. Thank you very much for your attention.
So we have again time for one question, and if not, then because we started a little bit late,
yes.
So that would be good already to pass to next to Angelo Rossa talk in three-dimensional conformation.